I'm Mehta. I am a professor of medicine, physiology, and biophysics, and I hold the position of Stebbins Chair in Cardiology at the University of Arkansas for Medical Sciences in Little Rock, Arkansas. My presentation is going to be about a very interesting topic: what happens to the brain after a myocardial infarction. It has been presumed that brain function gets disturbed. I'm not using the word altered, but there are certain disturbances in brain function after myocardial infarction. Unfortunately, it has never been shown, and if we don't know the mechanism, and we don't know how we would treat it. So we embarked on an experimental study in mice to look at the brain function after myocardial infarction. We took wild-type mice, anesthetized them, and created a myocardial infarct, and then closed the chest after ligating the left coronary artery. These animals were studied thereafter at 1 week, 2 weeks, 4 weeks, and 6 weeks after the myocardial infarction. As expected, we find that these mice had extensive infarct, and the size of the infarct became larger over a period of time. Then we isolated their brains and found that there was evidence of inflammation in the mice brain, especially in the cerebral cortex area and the paraventricular area. There was evidence for neuronal loss and neuronal apoptosis. This loss in brain pathology, these changes in brain pathology, were seen as early as 1 week after myocardial infarction, and similar changes were observed at subsequent time periods. There was no significant increase in the degree of inflammation or a reduction in the degree of inflammation over a period of time. Then we measured inflammatory parameters, mainly TNF-alpha and IL-6, in the serum samples of these mice and in the hearts and the brain, and found these inflammatory markers were increased very dramatically at 1 week after left coronary artery ligation, and then gradually began to fall. But even at 6 weeks, which would be like equivalent to about a year in humans, the inflammatory markers were still elevated significantly compared to baseline. Then my laboratory has been interested in a molecule called LOX1, and this molecule is actually a receptor for oxidized lipids, and we observed that the levels of LOX1 were increased in the hearts and brains of these mice who had undergone myocardial infarction. To determine if the elevated expression of LOX1 could be related to injury in the brain that I described earlier, we used LOX1 knockout mice. These mice have LOX1 gene deleted right from the very beginning. They're specially engineered mice, which my laboratory has created. And we observed that the LOX1 knockout mice, as shown previously, had significantly smaller infarct than the wild-type mice. More interestingly, the markers of inflammation in the brain and markers of neuronal apoptosis in the brain were very significantly reduced. The inflammatory markers in the brain were also reduced. So our data thus far shows that there is an intimate link which is between the brain and the heart, whereby a myocardial infarction through inducing systemic inflammation, causes inflammation in the brain and evidence of neuronal injury, which occurs actually very soon after myocardial infarction and persists thereafter. We believe that inflammation is the cause of this brain pathology after myocardial infarction, in which LOX1 overexpression may be very significant. Our next steps in this direction would be to look at these mice over a long period of time and to study the behavior patterns in these mice to see if myocardial infarction alters the brain function.